Hi everyone, welcome to the joy of botany, especially plant physiology. Let me introduce myself. I'm Kiran. I'm a 5-year integrated BSMS student of ISO Thiruvananthapuram. I found this is a great opportunity to talk to you about some concepts in the plant physiology. Thanks to you guys for that. Without further ado, let's begin.
In this particular lecture, I shall be covering different aspects of photosynthesis. I shall be talking about the definition of photosynthesis, followed by the site, that is the organelle involved in photosynthesis, the different pigments that play an important role in photosynthesis, and then I shall be briefly discussing the different phases of photosynthesis, that is the light phase and the dark phase, and towards the end, I shall be talking about the different important applications of photosynthesis. So, as the name signifies, photo meaning light and synthesis meaning preparation or formation of something. Photosynthesis is a process which actually involves synthesis of sugar in presence of sunlight in the green chlorophyllous tissue of plants. Actually, there are two phases. The light phase, where the light is helping in the synthesis of different energy related compounds, namely ATP and NADPH2, and that energy is utilized during the dark phase for carbon fixation and production of sugar. Definition: the process of photosynthesis is actually a photochemical process taking place in the green chlorophyllous tissue of plants in the presence of sunlight, where they procure light and of course carbon dioxide and water from nature, and they produce sugar or hexose sugar and oxygen as a byproduct. Of course, there may be another way of defining photosynthesis. We can say that the kinetic energy of photon particle is being transformed to potential energy in the form of energy-rich compounds, and finally it is stored as chemical energy of sugar in the green plants. It can also be said like it is the conversion of inorganic carbon from nature, that is carbon dioxide, to organic carbon, that is sugar, in the plant tissue. Actually, photosynthesis can be of two types, which is on the basis of release of oxygen. Accordingly, the process which releases oxygen is called oxygenic photosynthesis, and if it is not released, it is called anoxygenic photosynthesis. In oxygenic photosynthesis, actually the process is involving the green chlorophyllous tissue of plant, and it is also taking place in the cyanobacteria, where clearly there are two photosystems, namely the P680 and the P700, and the P680 is involved in the breaking of water molecule and release of oxygen, and the electron liberated from P680 through various carriers come to P700, whereas the electron liberated from P700 is coming to NADP and is reducing it. So in the process we get oxygen and also we get water. But in case of anoxygenic photosynthesis, which actually takes place in photosynthetic bacteria, that particular oxygen is not released, and it is shown by different bacteria like purple bacteria. And in this purple bacteria, what happens is there is a photosystem called P870, and it liberates the electron which is coming via a different carrier. Finally, it is received by NADP. At the same time, the electron that is replenished in P870 comes from H2S or ion compounds. So as a result of which, what happens is, in the process, sulfur or iron may be liberated. In another bacteria, namely chlorobium, there is P840 is the photosystem which receives the light, it liberates the electron, and this electron via FES and cytochrome B comes back to P840, but it is not directly coming back in the way it is received by NAD, so the electron replenished here also comes from H2S, and thereby here also oxygen is not liberated, instead of that sulfur is liberated. Comparison between oxygenic and anoxygenic photosynthesis. So, we can always compare the photosynthesis, which is liberating oxygen. Oxygenic and anoxygenic has some differences. It includes the photosynthetic pigment. Of course, the photosynthetic pigment in oxygenic photosynthesis will be chlorophyll, the different chlorophyll A, B, etc. And for that of bacterial anoxygenic photosynthesis, it should be bacterial chlorophyll. Of course, in this case, that is in oxygenic, there will be two distinct photosystems, photosystem 1 and photosystem 2. But in case of anoxygenic photosynthesis, there is only one photosystem, and the cyclic photophosphorylation is only operating. Here the electron donor in case of oxygenic photosynthesis will be water, but for anoxygenic it will be oxygen waste or other compounds. In the case of oxygenic photosynthesis, it will be producing oxygen and sulfur in case of anoxygenic photosynthesis. And the primary energy converting processes will involve the production of ATP and NADPH for oxygenic one, and for anoxygenic one only ATP will be generated. Photosynthetic organelle. Photosynthetic organelle, that is plastid. Actually, plastid remain as proplastid, which is a non-membranous structure which gradually developed into different forms of plastid, and this include the chromoplast, the leucoplast, and the chloroplast. Actually, the proplast, if it receives adequate sunlight, it will be converted to chloroplast. It will have chlorophyll, but if it is not receiving adequate sunlight, it will be converted to leucoplast, and it will act as a storage organ. On the basis of the nature of the storage compound, it can be amyloplast, which is storing starch. It can also be elaioplast, which is storing lipid, and it can be proteinoplast or alleronoplast, storing the protein or alleron grains. Similarly, chromoplast can be orange or yellow in color because it is having pigments of xanthophyll and carotene. Of course, the green chloroplastid is the most important organelle. This organelle is the site of light phase, that is in the grana, and the dark phase occurs in the stroma. But this particular organelle will have unique granule and intergranular region, and it will have DNA. It will also have starch granule and stromules. What happens is, with the gradual degradation of the leaf, when the plant gets old, then this particular chloroplastid is actually getting degraded, and this is degraded into red colored structure. This will be appearance of some membranous sac and carotenoid. There will be disappearance of chlorophyll, and gradually there will be stromules appearing, and the content will be getting decomposed, and the plant leaf ultimately will fall off. There is a unique process by which these particular plastids can be converted to other forms, like say for example, if it is a particular chloroplastid, the chloroplastid may be converted to senescent chloroplast, which I have already stated, but a chloroplast can be formed from chromoplastid, or a chromoplast can be formed from amoeboid plastid, which is unique. What does it mean? It means that the proplastid, through stages of conversion and interconversion, ultimately produces chloroplastid, which is the ultimate goal, and in between they will produce different other organelle, which is either chromoplastid or different forms of leucoplastid. But one thing is definite, that this particular chloroplast will always have this grana or membranous region, within which will definitely increase the membranous area and photosynthetic light absorbing service. But the chromoplastid, that way they do have pigment, but they do not have internal membranous structure. The chloroplastid itself can be of different types. There may be the chloroplast, that is the compound chloroplast, which is present in different forms of green algae, like in Chlamydomonas, the chloroplast is cup shaped. In case of Spirogera, which is a filamentous algae, the chloroplast is spiral, or it can be reticulated in case of another filamentous green algae, which is Oidogonium. So, this particular chloroplast structure is a compound one, and it will have multiple stud grain and protein stored in it, which are called pyrenoids or protopyrenoids. And depending upon the nature and the structure and shape of the chloroplast, the algae can be easily identified. Distribution of photosynthetic pigments. But if we come to chloroplastid structure itself, it is highly developed. It is unique discoid structure having a double membrane, and this double membrane structure is having an intermembranous space. Inside the chloroplastid, there will be the stacks of thylakoids producing the grana, and the grana are interconnected by lamellae, which is also called the stroma lamellae. What happens is, actually the chlorophyll molecules are arranged inside this particular grana in the form of chondrosome, and this chondrosome actually is formed of say 250 chlorophyll molecules or more than that. But more importantly, there is a unique structure resembling bacteria, and that includes the 70S ribosome and the DNA. The DNA within the chloroplastid and the presence of 70S ribosome, they do indicate that this plastid is not produced from the cell itself, but it has been acquired by some external bacteria, maybe because of the process of endosymbiosis. Now, when I say bacterial pigments, of course, it is bacteria chlorophyll C, bacteria chlorophyll D, E, and G. Mostly it is C, D, E present in different green bacteria, but bacterial chlorophyll G is present in heliobacteria, which makes them survive in high intensity of light. But when you talk about bacterial chlorophyll A and B, bacterial chlorophyll A present in purple bacteria, but bacterial chlorophyll B is present in Rhodopseudomonas and other forms of green bacteria. But when we come to higher plants, there are chlorophyll A, B, C, D and E. Chlorophyll A is known as the universal photosynthetic pigment because it is present in all plants and algae, that is green algae or other forms of algae, but not in case of bacteria. Chlorophyll B is present only in higher plants and green algae. Chlorophyll C is present in diatoms, dinoflagellates and brown algae, whereas chlorophyll D is present in red algae and chlorophyll E is present in yellow-green algae or in zoospores of Wucheria. Structure of chlorophyll molecule. The chlorophyll molecule actually is having two different parts, the head and the tail. The head is formed of magnesium porphyrin hydrophilic head and the tail is formed of lipophilic phytol tail. Actually, the head has magnesium at the center and there are four pyrrole rings attached to the magnesium by nitrogen. And to the fourth pyrrole ring, the phytol tail, that is the hydrocarbon tail, is attached. Basically, the second pyrrole ring is important because to this particular pyrrole ring there is the R functional group for chlorophyll A, which is the universal photosynthetic pigment. The R stands for CH3 or methyl group, but that particular chlorophyll B will have a R of CHO aldehyde group. So there is a basic difference between chlorophyll A and B in their structure, though the broad structure remains the same, that is the hydrophilic porphyrin head and lipophilic phytol tail. Biosynthesis of chlorophyll molecule. Chlorophyll biosynthesis starts from glutamic acid with the functional glutamyl tRNA and it is converted to aminolevulinic acid, and then with the dehydration of dehydratase enzyme it is producing 4,4-bilinogen and then it is producing hydroxymethylbilane and the urophorphyrinogen and coprophorphyrinogen and photophorphyrinogen. At this point, the photophorphyrin 9 is produced, which ultimately taking up magnesium and iron to produce magnesium photophorphyrin, which ultimately will produce chlorophyll and photophorphyrin 9. If it takes up iron, it will be producing phytochromobilin. Carotenoid pigments. Let me talk about the different types of carotenoids. Carotenoids can be lycopene, which is red in color, alpha carotene, which is yellow in color, beta carotene, which is orange, and gamma carotene, which is red orange in color. And the carotenoid also have a long chain with two hexagonal ring at the two terminal ends, and more or less the pattern is same for alpha and beta carotene. Only for gamma carotene, one of the chain is open. The carotenoid biosynthesis starts from glyceraldehyde 3-phosphate, which is producing deoxyxylulose 5-phosphate, and then gradually it will produce dimethylallyl diphosphate and geranyl diphosphate, phytoin, and then eta carotene, which undergo desaturation to produce lycopene, and then this lycopene will be producing two types of pigment. On one side it will be producing the beta cyclase action and it will produce the lutein, and on the other side it will also undergo cyclization to produce beta carotene, zeaxanthin, and in this way we can see that carotene will be converted to xanthophyll, which is yellow in color. Xanthophylls. Now let us talk about different types of xanthophyll. Actually, xanthophyll includes the lutein, which is yellow in color. Apart from that, there is violoxanthin, which is yellow in color, beta cryptoxanthin, which is orange in color, zeaxanthin, which is yellow-orange in color, and astaxanthin, which is red in color. In fact, these particular pigments are having more or less the same pattern of red or reddish yellow in color. Now, the xanthophyll interconversion is very interesting. It starts from phytoin, which is converted to eta carotene and will produce the red pigment lycopene, which is present in tomato, as you know. Under the action of LAW1 gene, it will be converted to beta carotene and alpha carotene on each side, and then beta carotene may produce the zeaxanthin, anthraxanthin, violoxanthin, and neoxanthin. These are all yellow in color, and on the other hand, alpha carotene might produce lutein and loroxanthin, which is reddish yellow in color. Energy transduction in photosynthesis. Now, let us talk about the energy transduction during photosynthesis. Actually, the photosystem 2, which is having the chlorophyll B or P4T, is giving the electron to lutein or P47D, then zeaxanthin then P480, beta carotene P500, then lycopene P510, and then chlorophyll B, which is P650. And finally, it comes to chlorophyll A, which is P680. So, electron is gradually being transferred from the second photosystem to the first photosystem, but in this process, they lose energy, following the second law of thermodynamics. But there is adequate energy present to liberate the electron from the photosystem 1, and thereby there will be photolysis of water or production of reduced NADP. So this is how we can see that energy which is not coming directly to chlorophyll, they are coming to other pigments, and these pigments are ultimately transferring it to chlorophyll, and thereby the photosystem is activated, and this phenomenon is called antenna pigment absorption. Photoreactivation center. We can see that the photoreactivation center is just like a funnel having different other carriers in the outside rim, and the chlorophyll remain at the center, which will gain all the energy. So in fact the energy which is absorbed by pheophytin, plastoquinones, cytochrome, they are all ultimately coming to chlorophyll, and thereby the chlorophyll is getting activated, and that will give rise to the electron, and that ultimately will switch on the cyclic photophosphorylation. Carriers involved in non-cyclic photophosphorylation. Photosynthesis is having two definite phases, the light phase and the dark phase. The light phase operates in the membrane of the thylakoid, where there will be the two photosystem, the photosystem 2 and the photosystem 1. The photosystem 2 will give rise to the electron, which will come to the photosystem 1, and the photosystem 1 will give rise to electron, which will be ultimately accepted by NADP. But water, which is actually undergoing photolysis, will give rise to H plus ion, and oxygen will be liberated out. But this H plus ion ultimately will be coming out via a specific ATP synthesis machinery, which is called ATP synthase, and thereby the ATP is produced. So we can see in this light phase there are three major end products, which is oxygen, ATP and reduced NADP. The last two, namely the ATP and reduced NADP, will be utilized during the dark phase. And the dark phase is actually operating in the stroma, which is having definite processes like carbon fixation, reduction, then synthesis of hexose, and regeneration of the starting compound, which is RUBP. This entire process is operating actually with the help of energy generated from the ATP and reduced NADP. Photosynthesis in the future. Photosynthesis has got some immense future. Actually, the production of hydrogen, which is the future fuel, or methane, may be the product of photosynthesis, and already there is enough research carried out in Finland where they are trying to utilize green algae photosynthesis to liberate hydrogen, and this will produce the future fuel. Apart from that, photosynthesis is also important in different other ways, which include it is the source of food, it is giving energy in the food chain, it is maintaining the balance between oxygen and carbon dioxide. At the same time, it is having enough oxygen to allow the other life forms to survive. Photosynthesis will also give rise to different plant byproducts which are utilized by mankind. And of course, the fossil fuel, which is the photosynthesis activity of the plant, is being utilized now by human beings in the form of natural gas, coal, and petroleum. So we can see that photosynthesis is immensely important in the present day life, and without photosynthesis, there will be no life in the earth. So, what we have discussed actually is the basic photosynthetic process. Definition of photosynthesis. We have also understood the different sites of chloroplast and their evolution in photosynthesis. We have talked about the different chlorophyll pigments and their pattern of their biosynthesis, that is how they are interconvertible synthetic process. I have just given an overview of the actual photosynthetic process, and towards the end I have also talked about the importance of photosynthesis.
That's all for today's lecture. We will definitely continue with very interesting and exciting concepts in the topic plant physiology. I wish you a good day. Thank you.